frustrating. And in addition, the genetics, really what they discuss in this chapter 9 is basic genetics. Doesn't really, I mean, there might be a few notes about microorganism genetics, but it's not all that different. So if you either you already had it in classes or you're gonna come across it again, I thought it's more interesting, and because we've worked on it before, it's more interesting to talk about why we use microorganisms a lot in biotechnology and the options you have using them and what they're good for. And like I said, there's the DNA fingerprinting is in this, the T-glow is in this, so you're gonna hear that again, and it'll help you, or you hear the main important points one more time, and then you'll probably have an easier time answering that in the lab exam on Thursday. So, as always, the preview. We'll just talk about the basic elements and applications. So what are the basic elements of, I guess, genetic engineering? What kind of characteristics of the DNA do we know to make them, to use techniques? Recombinant DNA technology, we talk about that. So you combine, and that's part of recombinant, that's in the word, so you combine genes from different organisms, and we've done that too. There's genetically modified organisms. Hear about those before. GMOs are, a big discussion, especially now with, I think the U.S. and Europe trying to do a big trading. They have a big trade agreement, or they're working on big trade agreements, and because of the Europeans, there's a lot of problems with that, because the Europeans think GMOs are should be off the table, literally, and here people don't. I mean, it's already implemented, so there's a big problem, because I heard a French farmer talk about how they're putting so much work in it, and they make so much, it costs so much money, and their food is so much more expensive, so if the U.S. comes in, they're going to lose everything. And it's hard to say, because a lot of times the consumer decides. So if they're still worried about what that could mean if you have genetically modified organisms, then they might not buy it. But you can imagine, for example, and we'll talk about those too. It's pretty interesting. If you look at the basic corn, what do you call the whole thing? Cob. And then look at it. I'm not from here. And then you look at it from when it's first, or the original, the wild type, just the amount of corn that you have on a cob now is completely different. And they're going much further than that. You now have modified organisms that, for example, they have a gene that prevents the formation of ice crystal if you get a frost. Things like that. It was just amazing that's already happening, and scary too, obviously, because people don't know what that means in our bodies. What are we doing with? How do we digest those genes or the DNA? I mean, what does it do in our bodies, et cetera, et cetera. And then treatment. When do we talked a little bit about that too before? But when can we use those technologies to actually treat disease? And then it could be in plants, animals, etc. And then we already did this fingerprinting and genetic testing. That's another, in the forensic world, another really important application. So what we skipped really in this lecture, and like I said, it's basic knowledge about genetics. It's the structure of the DNA, and then how you go from DNA to RNA, and how RNA is then used to produce the proteins in your cell, and that's really all it is. And there's a lot of there would have been a lot of material on just how what kind of enzymes are used in the process, and so that's something that's important. That we're not going to cover it today, but in general, you just know we have usually the double helix, and that contains all the genes, and then there's a process to produce protein from those, or to produce the proteins that the cell is going to transcribe and translate, etc. So that process we're going to skip because it's pretty general, or it's pretty much the same in microorganisms, except they have this circular, usually the bacteria all have the circular DNA. And so this basic knowledge is what when did they find out the exact structure? Sometimes in the 50s. So that was when the basic knowledge about the DNA double helix actually surfaced, and by now we're already applying that science to make products, to have all kinds of organisms produce all kinds of products for us, and we're trying to use it in medicine and other places. So the modification of an organism's genome is really what's done in bioengineering or genetic engineering. And then biotechnology is using organisms' biochemical and metabolic pathways. So if you think of the pathways of fermentation or pathways that certain organisms use, then that can be used to produce whatever product we're seeking, and so that is in biotechnology. So those are, the engineering is probably more the putting it together part, and then the biotechnology is using the pathways and getting a product out of it. So in the beginning, we'll just talk about what the principal properties are of DNA and what we use it for. So what they found is that even in a test tube, but in nature, basically if you go close to boiling, the two strands of DNA separate. So the hydrogen bonds, remember, like there's all the nucleotides, so you have adenine, guanine, cytosine, and thymine in DNA, and there's the nucleotides have a phosphate group and a 5 carbon sugar, ribose. And that's the backbone of it, and then this double helix is the DNA, and so that helix can actually, those hydrogen bonds between those bases are actually breaking when you heat the DNA up to from 90 to 95, or around 90 to 95 degrees Celsius. If you want to visualize that again, just what the DNA looks like, because you might run across, or we're going to come upon that a few times, then just go back to chapter 2. Remember, I don't know if you've already looked at it, but that's one of the macromolecules, and then you'll know what it looks like. So when you heat it up, they separate, but then actually when you cool it down, they get back together. So it's the hydrogen bonds form again. So it's not destroyed if you don't go higher. And don't imagine it as a process where the exact places, the DNA, say you would heat up the DNA of one of your cells or multiply it, but your cellular or your DNA that's contained in the nucleus, and you would heat it up, it wouldn't just do this and then that. You wouldn't just have exactly the same. I mean, it's just looking for complementary, for areas that fit, and so that it gets back together, but it's not necessarily exactly the same. But to know that we can actually separate those and then have these open sites, these open bases where we can actually put say pieces of DNA that it binds, is that's the technique, or that's the basis for a lot of technologies. So it becomes double-stranded again, and so if you're, for example, if you say, and we'll get to that later, but if you say, okay, I know E. coli has a specific sequence, and it's 6 long, and it's just a combination, say it's a G, T, A, T, G, C, for example. Say that's pretty unique for E. coli. You could then do this with an unknown sample of DNA. So you take a patient's sample, you extract the DNA, you do this, and then you throw these in. Just imagine like that. I mean, it's a little bit more complicated, but you put all these short pieces in, and then you try to detect where they land. If they find places to attach, then if that's really, really unique for E. coli, then you could identify that that person has E. coli DNA in either in their cells, so you have an infection, or you have at least those bacteria in the body, and it's just there's a lot of different ways to use the system. Is what I'm trying to say, and you can detect microorganisms. Yes.
Are the sticks they're looking for usually that short?
Not really. I mean, I think when we come to those two, when you use, for example, primers, which are you put in a solution of single-stranded DNA to start a process of making the second strand, which we'll get to too. It's probably 20 base pairs. So I would imagine they're longer pieces. You really look for longer ones. But at the same time, if they get too long, I think there's more mistake. The probability of mistakes are higher. But a lot of times, you really, although there's only these 4 bases, a lot of times you find that there's specific sequences in the genome of an organism. You can actually really look for those or detect those. But they're going to be longer, most likely. But if they get too long, you're going to have more error. So most important, there's 3 enzymes you should know. So far it shouldn't be too complicated. It's just how some of the basic characteristics of DNA are used. So there's restriction endonucleases, and you've already seen those. So what those are? Does anybody? Well, it's written down here. Remember when we used restriction enzymes? So when we remember your gel, you basically we didn't apply them, but they were cutting the DNA. Remember I showed you how they have different areas. So that's one of the important ones because of what you did, for example, looking at the size of the DNA's parts after a specific restriction enzyme was applied. So they originate in bacterial cells. I told you that when we did the experiment, and they recognize a specific sequence of DNA, and that's when they cut. And we usually know in plasmids, or we usually know when they do. Okay, 15. The last one. Sorry. I told you. The last time I forgot complete is like I got a quiz back like at the end of the lecture. Like, where did that come from? So do your last one because I got a thing in. I don't want to think about this anymore. So they thank you. They splice or they cut the DNA in a specific place, and
what was the name of the enzyme we used?
There were was it ECO1R?
Oh, that's
one of them. HIN3. Oh yeah, here they are, HIN3, and then this one we didn't. And so a lot of times they cut like this, so they find what they call palindrome-based sequences. So if you look at that, they basically find areas and then they cut them usually in different places, but those are, if you see here's huh? And sometimes they cut straight. So sometimes they cut like the last one, I can't remember what they call that one, but in general those are the cuts. And so that that creates these single strands over here and then I'll show you in the next one. But what they call those sticky ends, I think, and that's and then what those enzymes do is they basically make restriction fragments. So you have all these fragments of DNA after you apply this restriction enzyme. And because our DNA sequences vary, even in the same species, you see a lot of different sequences, especially because not all of our, there's a lot of DNA in our genome that's not really coding for anything. It could be left over through evolution. They're not quite sure exactly what most of them do, but there's a lot of DNA that's not translated into protein. So there's for example repetitive sequences that go on forever, and so in those you might have, they call them macro and microsatellites, but that's not that important. But what you should know is, so those could be 17 sequences long or 17 units long. Say you have a mini microsatellite that just looks like GTG, for example, and you would have 17 repetitions, and then I only have 14 repetitions. It doesn't matter because it doesn't code for anything, but the number of repetitions is different between people. And so if you find a restriction enzyme that cuts on those, you can basically say, oh, in my case, you know, if you have that, and it cuts here, and it cuts in the next one further down, my base pairs between those cuts, my fragments are 200 base pairs long, you're 250, that's how you get those lines. And so these differences in the cutting pattern give rise to fragments of differing lengths, and then that allows the comparison of DNA of two organisms at a specific site. So we can look at those repetitive sequences and then determine, you know, for example, is this DNA from the same person, things like that. So that's in forensic an important technique. So we have the enzyme, the restriction enzymes, really important, and we use those. So don't forget those. Bacterial, they cut the DNA, and then ligase is one that uses these sticky ends. I'll go right back. But so now we have these sticky ends because if you have this cut of the DNA, and they rejoin those bonds that are cut by these restriction enzymes. So ligase is one of them, one of those enzymes. So that's the second one. The restriction enzyme and ligases. And so used for final splicing, basically meaning for final introducing and connecting genes of different origin. So if you have, for example, a plasmid like we did, you cut them, and I'll show you that here. So for example, you're interested in a specific gene, and you know that you can cut out that gene by looking at specific areas. So you say, I'm interested in a specific gene, and I can cut it in two different areas because I know there's a restriction enzyme that cuts around this gene. So you cut it out, and then the ligase would basically, if you cut the plasmid with the same ones, then it can, in a lot of cases, it's not 100%, but it can actually connect these sites that are cut by the same enzyme, restriction enzyme, and that connection produces a plasmid that's possibly expressing the gene that you're interested in.
So they're used kind of in connection to
Yeah,
each other.
yeah. And then the last one is, and you've heard this one before, I think, reverse transcriptase. So that makes a DNA copy of RNA. All you really should know here that, and we've talked about the reverse transcriptase before in viruses, because a lot of the viruses have RNA.
Did you say this is the third one?
Yeah, that's the third enzyme that we'll talk about. So if you have an RNA virus, that means the genetic, viruses can have DNA or RNAs. So RNA viruses like HIV have basically their genetic code is coded in RNA, and so reverse transcriptase is then translating it back to DNA, and it can then be read. And so what's important here is that, like I said, there's a lot of, and that's probably good to see that here. If you look at the eukaryotic DNA, if you look at our DNA, there's a lot of introns, and introns are areas that are not, they're not transcribed. There's no information in those. But then if you have RNA, usually what happens is they're taken out. So the RNA is everything you need. So if you have a lot of garbage in your DNA that's not even used for anything, or say old, what do you call it, empty weight, something like that. So you have a lot of DNA in your genome that's not being used, that's being cut out. And so the reverse transcriptase can be used. If we use this enzyme, you can go and make, out of the RNA, which is all the important information, you can make cDNA, which is all the DNA that codes. So that means you can, for example, look at differential gene expression by that. But because you're gonna know, if you use the system, you can basically in a cell you would be able to say, I know what RNA is in the cell, so those are the proteins. Those are the proteins the cell is gonna produce. And if you can detect them with that complementary DNA, then you would know what the cell is making. It's not all the information, it's just the information that the cell is currently producing. And that's what you can use that for. But that's the least important for right now because it's a little bit more complicated. Just know that there's these three enzymes. So why you use all these techniques, for example, is because if you want to put this information into a bacteria, for example, the bacteria can only read the DNA a certain way. It has to have double-stranded DNA in it. So if we, for example, wanted our bacteria to produce green fluorescent protein, it would only work if the bacteria gets a plasmid that has the right DNA in it. If you put a gene in that includes introns and other things, it might not make what you want it to make. So you basically go through a process to deliver the right DNA to the bacteria. And that one is, most of this is not important, but for us right now, but you basically go from the DNA to the RNA, which means all these introns that are not important are cut out. So now all you have left here in this orange is the information to make a protein. These not used areas are cut out, and then from there, with reverse transcriptase, you can make cDNA, and then you can use that double-stranded DNA after that process to put it into a plasmid, for example, and then the bacteria can read it, and the bacteria can produce the protein. So that was just looking at what's the DNA important, or what are the characteristics that are important to us for engineering, and then the three most important enzymes that we use to produce, for example, plasmids, or to produce DNA that we then can transfer into organisms. And this is just there's a few slides about how we analyze DNA, and obviously all these techniques. Sorry, yeah, that's one of them we did. All these techniques are connected, so it's not that you would, you analyze the DNA. You probably have done some of the other things we just talked about before. So gel electrophoresis is separating DNA fragments based on size, and we already done that. So remember that the size was important. Which ones, well, what direction does the DNA usually travel towards, positive or negative? Positive, because it is negative. And that you can, you know, if I have this blurry image of, there's these phosphate groups with a negative charge on each of these nucleotides, so that's why the DNA is negative. If you go back to chapter 2, and then if you have these bands, which the furthest travel is the largest or the smallest? Smallest. So it gets through these channels the furthest. So you cut your DNA with restriction enzymes or endonucleases. You get these pieces, and then you apply them in these wells, and then they travel. You stop the travel, which, you know, did anybody? I can't remember. Did anybody watch the dye go all the way to the end, or did we just say half an hour? Yeah, I was thinking that would have been next time I would run it longer, because they're pretty. Some of them are pretty nice, but you could run so much further out. We didn't do that. So negative charge of the molecule causes the DNA to move towards the positive, and the negative charge comes from where in the DNA? Perfect. Okay, don't study anymore. Then we did this one, and then we stained it for observation. So you apply a stain, and the stain, because we all, you know, in the beginning everybody thought we're gonna see this. You know, we looked at it and we saw the dye, but the dye is just an indicator of how fast the DNA is traveling, at least the small parts. But then we actually stain the DNA. So you find a stain that attaches to the DNA, and that's what you saw on the UV light under the regular white light. So you can look at DNA fragments. You can look at genetic similarities, and we talked about that enough. That's the same thing here. And so the only thing we didn't do is you usually add a marker to a side. And I don't know why they have no markers here, because markers are the easiest thing to get. But you know, this marker would say the pair you see here is 5,160, I think, base pairs long. And if it's down here, it's only 1,000 base pairs long. And if it's down here, so you can actually look and say, I got the right piece. You know, you would, for example, one of the steps along the way, if you want to cut out a gene, you would say, I'm not sure I did the right thing. I don't know if the enzymes worked, they were old in the freezer, whatever. So you apply this method first, and then you take a little part of this. Say you want to later on put that somewhere else. You want to use that DNA in some kind of system where it produces a protein, but that comes later on. You could run it on a gel and just say, did I cut out the right piece? And then you put a marker in that's close to the piece size you imagine. So you can go to, online you can find these, you can look for any sequence, and so you would know the length of my segment should be, DNA fragment should be 250 base pairs, and then you find a marker that is about that, and then you can see, do I get a band there or do I not? And then if you do, you can use the rest and keep working with it. So it's kind of like a control mechanism, or used as a control mechanism also, because you're working with things you don't see. So to be able to run them here and see how big the fragment, the DNA fragment is, allows you to know, did I cut out the right piece? That's just one example, but it's definitely things they do. Another thing we didn't do is hybridization, and all that means is, or what that means is that a single-stranded DNA, and that can happen through heat, can actually unite, which we've seen too when you cool it down, either with its own again, so the two single strands come back together, but you can imagine it also attaches to single-stranded DNA or RNA, and RNA can actually reunite with RNA also. So there's a lot of options of them after you heat them up, and then you add, for example, pieces, like I said, you add this E. coli piece that you're looking for, and add that, then they hybridize. And so what happens is when they hybridize, or what that's used for is what I showed in the beginning. It's that you do use short DNA fragments, and those are gene probes. So you have a known sequence, and you think if the complementary sequence, so if the right DNA is in my sample, then it should attach, and then there's different ways of making that visible. So you have you have unknown DNA in your sample. You have that single-stranded because of the heat, and then you put a high concentration of a probe in, of a short piece with a known sequence that you're looking for. And if it actually attaches to that DNA, you know that that DNA is the one you were looking for. You can be pretty certain. And so one of those, I'll show you quick. I've done a lot of what they call Western blots. This is a Southern blot. I guess the inventor of this one, which was the first one, was called Southern, and then they made all kinds of other blots, and then they started calling them Northern and Western and what's left? Eastern. There's no Eastern blot so far. So if you have any ideas, you can get the Eastern blot on your list. So this is and I'll show you in a sequence. It's in your book too. That's a way to make those attachments of probes to DNA visible. And so you can, if you apply this technique, you can, for example, diagnose a pathogen in patient DNA. And so in this case, what you do is you cut just like we do, or we did. You cut the DNA. We didn't cut the DNA, but we got the cut DNA and applied it. So you would just use DNA. You cut it with restriction enzymes. So you have all these fragments, and then you let them run on the gel. So you have all these fragments all over the gel. And then the interesting part is what they now do is they kind of try to want to get this DNA out of the gel onto something they can work with. And so what they do with this whole really pathetic technique, because I remember doing this thinking like, are we in a molecular lab, or are we I don't know, in the back of a restaurant washing dishes, or like applying some kind of technique? Because there's all these paper towels you use, and people use all kinds of materials, and then there's a weight block, and they use whatever. So it's all it is really that you have your gel in here, and what you're trying to do is you're trying to get buffer, or get, a little bit different than water, but mostly water, to get out of this and basically take the DNA from the gel up to a DNA binding filter. So there's just like a filter paper. There's a paper, and that's above the gel, and then through capillary action of the buffer, that DNA comes out of the gel and is carried onto a piece of paper. And when you're done with the Southern blot, you have a paper that is actually the same thing as the gel. The gel lost the DNA, and the paper has it now. So you just transferred it from the gel to the paper. But you do that because you couldn't run the DNA on a gel, on a paper. How would you separate it? So you separate it in the gel, and they're like, how do we get this onto paper? And so how they do it is they have this buffer carry the DNA over to the paper. So now it's glued or stuck to the paper, and then you put it in you put it in a fruit bag, Ziploc, but it's still happening all the time. I mean, that's how it works. They're all, people who came up with this are pretty smart, but they use their lunch bags to get things done. So then you put it in there, and then you put your probe. So these pieces of DNA that you want to look for, you put those in there, and then they hybridize. They find the complementary DNA and attach. And so now they're attached together, and then the probe is usually radioactive, like very lightly radioactive. And so what it does is, if you then have this filter paper and you overlay it with film and expose it for a while, the radiation from those bands, from those DNA bands that are now radioactive because the probe was radioactive, makes these lines, and then you have basically you then can read which DNA has hybridized with the probes, or if they have.
So this is not, the gel isn't already dyed then?
No.
You don't dye the gel?
No, because you have one where you dye the gel just to see what you have in there, but what it really does is it allows you to hybridize these pieces, or to connect these radioactive pieces that you know the sequence of to unknown DNA, and then if they hybridize, you wash it all off, and then whatever stays on the DNA, meaning it found its complementary piece, is then gonna give you a signal in form of a basically a band that you see on the film.
Why would you want it on film instead of in the gel? Why would you want it on film instead of in the gel?
Well, it's just a way to make it visible, because you would not be able to. You can't
see it on the paper.
No, you wouldn't be able to see it on the paper. So the probe, you could. That would be an interesting question. Why don't you, for example, have a probe that has another color, and then you could see it directly on, and maybe that's already in progress. I don't know, because a lot of times they're able to put color or dyes, attach those to the probes, but
it wouldn't take the dye with it if you use the dye gel.
Well, the problem is if you do it in a gel, there's the way these probes attach to the DNA, that only works in a specific, like they do it in here and it wouldn't work in the gel. Like there's no way of connecting those probes to the DNA after they ran. So the way you do it, you expose the DNA to these probes by having them on a filter, and then you make them visible. So that southern blot, that just means you're hybridizing, and you're also hybridizing in situ, and that's called fish fluorescent in situ hybridization, and it just means you can actually hybridize probes to intact cells. So in this case, it's hard to see, but you see that a probe that has a dye to it can actually get into the cell and find complementary DNA. So use that for diagnostics. For example, if you have a virus infection and the virus is able to have its own genome integrated to the host genome, you can find that. So don't worry about it too much. It's just another way of looking at, or another option to use hybridization techniques to find something. So all you should remember there is what that helps you with is getting DNA, or finding the DNA you're looking for by using little detectors, and those are the probes. So if you know, if you have a sequence, and we'll look at that in a little bit, if you have the sequence of an organism that you know, you can try to hybridize it to unknown DNA and say, is this genome in my unknown DNA, or it has the virus, for example, infiltrated this cell, because there is a counterpart to it. And so one of the steps to get to these points, one of the steps is, or to start this whole process, is the sequencing part. And so what that means is really, what is the sequence of a gene, and then what is the sequence of your genome. And it used to be absolutely, I mean, I don't know how long they worked on the Human Genome Project, but to just find out, which is a huge amount, but over years, they were trying to figure out what are all the different, what's the complete genome of the human species, what is its what are the sequence. So they have, I don't know when they finished, it's a while ago, but now you can find every single base or nucleotide and how in which order it is of the human genome, and they do that pretty quickly now for other animals. I mean, it used to be so difficult, and then a few years ago, I remember somebody said to me, would you be interested in like having one of the animals you work with completely sequenced because this company is doing a promotion? And like, what? I mean, that used to be, like, it felt like the whole nation was trying to get this done, just like, traveling to the moon, and now it's some company wants to get your attention, and they quickly sequence the genome of some fish or whatever you're working with. So it's gotten a lot faster and easier, and they made progress throughout the process of sequencing it. But what it really is what's the actual order that of all the bases that are in your genome. And the Sanger technique is one that used mostly, and I want to show you just so you've heard it before. It's not important that you know all the steps. But what you do is, so you have, say, you could again take a restriction enzyme and cut out a piece, because you can't, it would be pretty hard to do. It's usually done in smaller segments. You can't just go and say, oh, I'll take your chromosomes, I'll denature them, and then you have all these open spaces, strands laying around, and we'll just do it in one piece. So you have to cut them up, and it's a long process, really complicated in terms of afterwards fitting those all back together so it makes sense. But you cut them, and then you have these, and then you denature. So now you have single strands laying there with their open bases, and they can be, and then you try to synthesize the complementary strand onto it. And so what you do is you use 4 different tubes, and they have primers, which means there's some beginning, some piece attaches, and it uses usually sequences that are very common. So you have a primer in there. Just remember, primer starts the process. And then you have DNA polymerase, which is going to be able to add these nucleotides, and then you put all nucleotides in, so they're all available, but you put them in single ones, so you put nucleotides in so it can add them. So you have the whole process, the primer, the DNA polymerase, and the nucleotides are giving you the option, or giving this reaction, the test, to all the ingredients to start making the second strand of DNA. But then you also add nucleotides that are not able to, they're basically stopped nucleotides, and it makes more sense if we go there. So you start off with your regular DNA, you denature it, now you have a single strand, and then you have a primer that attaches, and now in these, so the primer attaches, and now the DNA polymerase is ready to build this. And what happens is the T is looking, this is a complementary for an A, so some A has to move in here, and you have these nucleotides in here. So an A moves in, and then a G, and a C, and a C, and so it adds it to it. But there's also in the mix, which is a really, really smart idea how they approach that. There's some in that are called, where are they? In this case, they're basically the ones that are a small amount of single types, so they don't allow more connections. So they're like a stop G, C, A, T. Usually when this nucleotide adds, you can add another nucleotide, so it makes the whole row. But you add a few, or you add a certain concentration of some of these nucleotides that do not allow more. Or the process to continue, and they're labeled. And so if you label them, say, yellow, blue, green, and red, and eventually, all these processes produce these, or go into the process, at some point you always get the stop one.
Wait, so are you saying you put in, like, for example, way less of the G, so you know that it would stop there?
No, less, you mean?
Yeah.
no, you have a certain concentration of these regular nucleotides, and they could go on forever. So they would just do it, and then every single time you would look at it and you say, okay, I have no idea what the sequence is because I get this back basically with all these different colors.
Okay.
But if you add some that are basically attaching here, say this one, this A attaches, and it cannot add, this is, instead of a phosphate group and everything that it usually has, say it has a dye on it, and so it attaches and it prevents the polymerase to keep going, then you have a green signal here, and then on the next strand, because you've got millions of them, it goes normal until this one, and then this yellow one stops the process, and then here the blue one stops the process, and then you add all four together and run them on a gel, and because this one is the shortest, you get a green signal in the beginning, and then it was the second shortest, and you get a yellow one, so that those pieces of nucleotides are put on a gel and all ran together. And so if you look through the process, it's a little bit complicated to understand immediately, but you then have the shortest nucleotide that you produced has a green ending, and you see green first, which is this one, and then you see one that goes a little bit further and it's yellow, and a little bit further, and so that's how you then read the sequence. Does it make any sense?
Yeah.
And the thing is, it's all automated, so it's not, you don't have to, it sounds complicated and it sounds tedious, but it can work because it's now automatically done, so it's a lot more precise than we would probably do it. But that's how you would get a sequence of a certain area, or you can.
Because how long are these sequences then? They're like thousands and thousands. Huh?
Yeah, but you would probably take short pieces, and then that's what I'm saying. They try to then figure out how to put them back together, and where does the last one stop, and where does the new one start, and it's a lot of work to then put these sequences that you get together, because they sometimes overlap, and there's all kinds of difficulties with that. But you kind of get this area, and so I think, again, after a certain length, it becomes problematic also. So you would take a sequence, maybe and said, can you sequence a gene that should be 250 base pairs long? And they would probably be able to do this. I don't know what the numbers are now, but I remember that what used to be the case, maybe around 200, 250, and then you would basically go do the first piece, and if you need more, you have a primer that attaches further down the road and sequence that, and then you put all these sequences together. But what you really should remember is, the sequencing is not so hard anymore, so we can now pretty easily find out what the whole genome does look like, and then we can say, oh, if I have an organism that's very specific for a certain sequence, or I find a very specific sequence for an organism, I can use that to detect it, or I can use that for other purposes. But that's for pathogen, that's important. And that's just a quick summary of what they do. So the genomics, if you ever come across those, the genomics is the study of organisms' genes and the functions. So that's what we've talked about. And then there's proteomics, which means they look at the proteins and the functions of the proteins. So there's a lot of different, like Western blots instead of Southern blots. You do the same thing, but you run proteins on a gel, and then you transfer proteins, and then you identify proteins. And I think I told you before, say heat shock protein was something I worked with. So they would say, well, we want to know if an organism is stressed, and they know that this certain protein is expressed if the organism is stressed. So you probably all have it transcribed right now. It just increases in concentration, and so you would do a Western blot out of whatever tissue you're looking at, and see if that specific protein is expressed more or less, and you look at the band. So you would run it on a gel just like these, and it would run down, because proteins you can run on gels too, and then you identify them and you look at the bands and how strong they are, and if they have a really strong band, they have a lot of protein in them, a lot of the protein you were looking for. So it's the same kind of technique. It just looks at proteins instead of DNA. And then there's other ones where you have, you look at the genomes of all the all the organisms in an ecological niche, and that's called metagenomics, and metabolomics is another one where you actually look at all the chemicals that are happening in a cell. So they're going to extremes. If you think about how hard it was to figure anything out, and now they're trying to figure out what all the different chemical compounds or all the compounds you find in a cell with this technique. So I have a few more, and then we can just go through, after a short break, we can just go quickly through the rest, which is where are those things applied in health, where are they applied in food production, and then that should be it. One other thing you've probably come across a lot of times is the PCR. I just heard it. People do PCR, hopefully. Maybe not, but it's Irene is the lab technician, and she used to work with this guy who came up with this idea, and he wrote a book. And I think he really wants to be a hippie surfer, and came up with this. And then what you see on his book is he's standing there with his longboard or whatever on the beach. Like, well, what's that got to do with your PCR invention? But I think it's more important to him that he's a surfer than that he came up with this. She said he's a pretty crazy guy, but he was, as he describes it in his book, he'd go on vacation and then just think about this stuff until he had a solution. So he wouldn't even do it in the lab. Irene said he was horrible in the lab because he would just messy and never really knowing what's going on, but he was brilliant in his head, so he would just go on trips and then come back and be like, I think I have it. And this one, I think he got the Nobel Prize for it. It's basically, and you've already, you've already seen this system, and so the only difference is, or what this is really good for is to make a lot of copies of a certain piece of DNA. So if you have DNA evidence from a crime scene and there's not much, you can make as much as you want. You copy it until you have enough to work with. That's really what PCR is good for. I know that I had an anthropologist person, it was a professor in Munich, and she would always dig out bones, like from these crazy old sites where churches used to be from the 13th century or 14th, and then they would do PCR to basically whatever DNA they could find on the remains of people, they would try to amplify it to get as many copies as possible, and then look what has changed, things like that. So it's just remember PCR meaning you have a chain reaction going on that amplifies or makes a huge amount of DNA, and then you can do whatever you need to do with it. And this is a terrible slide, but and I don't want to confuse you, all you do in this case is you use your DNA, say you have a small amount of DNA from a blood drop or from a little bit of blood that you find on a crime scene. So you're able to isolate and separate and or isolate and concentrate that DNA. It's not much. You don't know what, it's not enough to work with. So you denature it, it opens up, you put a primer on it, and then you supply all these nucleotides, just like before, and they attach, and then you denature again, so you have new strand. Now you've done this. So after opening it up, it basically produced the complementary side of it, and now you have a double strand. And the double strand is again heated, so it opens up, it does the same thing. You have a new strand. You heat it, it opens up, it does the same thing, it produces the complementary side, and so you go from one fragment to 2 copies to 4 to 8. So if you run this, they say for 20 to 40 cycles. So cycle is heating, production, cooling, heating, production, cooling, and they have cyclers. They have little machines like this big now. They used to be like as big as this room, and now they're like little benchtop PCR machines. if you run it for 20 to 40 cycles, depending on how much you start with, you get millions of identical DNA molecules. So he came up with this idea, and that's not too old either. Now it's in every lab, but he came up with this idea to said, well, if you know, if you can sequence, or maybe they knew, you know, it denatures, how could we make this work that, you know, it would automatically produce a lot of DNA? And probably initially they said, well, we'll denature it, we produce all this DNA, and then, oh great, now we have 2 instead of 1. And so to go to the next step and heat it up again and do the same thing over and over and over, probably required a lot of tweaking how to do it in terms of the chemicals, but now it's a technique that's constantly used for everything. And Irene was saying that she worked in a lab, I think with him, and she switched, and then this other company she worked for wasn't interested. They're like, that's not going anywhere. Like, they heard about it, and they invested in something completely different. And then, I mean, now whoever bought the, I don't know if it's, what's the name of it? Yeah, patent. Well, whoever bought that is probably not working right now. So, what is it right now? 22. Yeah, maybe we'll take a short break, and then if we go to the last, whatever, there's maybe 18 left, but they're pretty quick because it's just what we use these techniques for, and then we definitely have half an hour, 45 minutes left to talk about Wednesday and Thursday.